Biology. The word bio means life and ology means study. The study of life is known as biology. Life originated on Earth was several billion years ago, and they all originated from single subunits called cells. Cell. It is the basic structural and functional unit of all living organisms. Cells can be of two types, which is prokaryotic and eukaryotic cells. Prokaryotic cells. These are small, simple, single-celled organisms lacking a nucleus or membrane-bounded organelles. Examples are like bacteria. Eukaryotic cells are larger, complex, and contains a defined nucleus and organelles. Examples are like plant, animal, and fungal cells. Cells are extremely small and they cannot be observed by the naked eye. Therefore, to observe them, we use special instruments like a microscope. Microscope. It is a device which is used to see cells at a microscopic scale. So, in today's video, I will show how to make one prokaryotic cell like bacteria and two eukaryotic cells which is a plant and animal cell. So first, I will show how to make a plant-based slide, which is an eukaryotic cell. So for this purpose, the specimen that I'm choosing here is a piece of onion. So I dissected the onion using a knife from the dissection kit and peeled out a thin plastic membrane using forceps. I placed it onto a clean glass slide and added some methanol to fix it. Then I cut the specimen into the exact dimensions that I needed and let the alcohol dry off. Then I placed it for staining. This is because most of the biological specimens are transparent and colorless. So we stain and color them in order to see them more clearly and observe them more better. Any stains from the following can be used for staining. Methylene blue can take up to 5 minutes. Crystal violet can take up to 2 minutes. Saffron can take up to 3 minutes. However, in this case, the stain that I'm choosing here is methylene blue. So using a micropipette, I pulled out some stain and applied it onto the slide. I waited for 5 minutes and then washed it using some water from the wash bottle. Then I let the excess water dry off and place a very small amount of water onto the specimen. Then place a cover slip onto it and remove any excess bubbles from the slide. Now the slide is basically ready, so I placed it into the microscope. At 5x we can see all of the tissues of cells. At 10x we can see each and every cell individually. At 45x we can see each and every cell having a nice much more detail. We can see the cell wall, which separates the cell from the other cells, as well as it also provides a fixed rigid shape to the cell. We can see the cell membrane, which is the inner layer of the cell wall. It keeps all of the contents and organelles inside of the cell. However, the cell wall is not present in animal cells and it is present in plant and fungal cells. The dark stained mass is called the nucleus, which contains the genetic material and is the code of life. The cytoplasm is the fluid on which all of the organelles float. It also helps in transport of materials and take out reactions into the cells. Well, this was all about plant cells, and now let us talk about animal cells. So, as I said before, animal cells are also eukaryotic cells consisting of various tissues and comprise of more than one cell. Us as humans, we are also comprised of various types of tissues. Mainly, there are 4 types of tissues in our body: epithelial tissues, connective tissues, muscular tissues, and nervous tissues. The study of tissues under the microscope for detection and diagnosis of disease is called histopathology. So for this purpose, the specimen that I'm choosing here is a drop of blood, which is a connective tissue. This is because observation of blood cells is quite common at clinical pathology and at my grade of level. So to do this, I took out a lancing pen and a lancet and I charged it with a needle. Then I sterilized my tip of finger with about 70% alcohol pad. and I placed the pen onto my tip of finger. I pressed the button to prick my finger, then I applied pressure to the near side of the prick. After this, act quick as the blood will start to clot. So I took out a micropipette and took some blood. Place a drop of blood onto the end of a slide. Then place another slide onto the drop of blood and smear it to the end. The specimen that I made over here is not proper, and a proper slide usually looks neat with a head and a tail. This is maybe because I suck at making this and it requires some proper form of practice to make a proper smear slide. Let it dry and fix the slide using some methanol. Let the methanol dry off and then place it for staining. For this purpose, the stain that I'm choosing over here is a Gemza blood stain. So I did make a 50-50 stain and water mixture by adding 1 ml of water and 1 ml of stain into a long tube. Then I shaked it to properly mix it, and then I poured it all over to the slide. Now to let it stain for about 15 to 20 minutes. After 15 minutes, I washed the slide and dried off the excess water. Now I placed it into the microscope, and all I had to do is to observe. At 5x we cannot see much more clearly, but at 10x we can see all of the blood cells and tissues. At 45x we can see each and every cell with much more detail. We can see the RBCs, which are called the red blood cells, and it is responsible for carrying oxygen all around our body. We can also see some WBCs, which are called white blood cells, which fight against pathogens in our body to protect us from diseases. Here are platelets, which help in blood clotting. There is also another component of blood that I didn't mention here, which is called blood plasma. But if you want to learn more about blood, then let me know down the comments. Well, this was the procedure on how to make an eukaryotic cell, which is a plant-based slide and an animal-based slide. And now let us talk about making a prokaryotic cell, which is called bacteria. Bacteria is a unicellular microorganism which exists, and it has an important part in our ecosystem. There are mainly two types of bacteria, which is a gram-positive and gram-negative bacteria. Examples for gram-positive are like Staphylococcus and Streptococcus. Examples for gram-negative are like E. coli and Pseudomonas. For this purpose, the specimen that I'm choosing over here is a gram-negative bacteria, which is E. coli. E. coli is a rod-shaped gram-negative bacteria, and it can cause infections like UTI, also known as a urinary tract infection in humans. To do this, the bacteria is already grown on a cultural medium. So using my inoculation loop, I first sterilized it by using some heat. Then let it cool down. I took a small colony of E. coli and placed it on a specific spot of the slide. I then sterilized the loop, and since the bacteria is waxy, I fixed it using some heat. Then I placed it for staining, and now all I had to do is to add the following stains. I first added some crystal violet and let it stain for about 1 minute. Then I washed it and added some iodine solution for about 30 seconds. Then I removed the iodine and added some decolorizer for 30 seconds. After this, I washed it and applied some saffron for about 1 minute. After this, I washed the slide and let it dry off the excess water. I then placed it into the microscope, and all I had to do now is to observe. At 5x we cannot see much clearly, so at 10x we can see the colonies of the bacteria at very specific spots. At 45x we can see the proper rod-shaped gram-negative bacteria called E. coli. Well, this was for a gram-negative bacteria. So for a gram-positive bacteria, I placed an already prepared slide called Staphylococcus or Staph aureus, which is a gram-positive bacteria. Now at 45x, we can see the round spherical cocci shape of Staphylococcus under the microscope. In general, gram-negative bacteria gives a nice pink or red color by gram staining, and gram-positive gives a nice purple or blue color under the microscope. Well, this was all about bacteria in general, and if you want to know more about it, then let me know down the comments too. Well, this was all about making a prokaryotic cell. And if you like watching videos like these, consider subscribing to my channel and liking my videos. And I will hopefully see you in the next one. Thanks for watching.